So the question was sort of how good a publication record do you need for the
Two
compendia?
publications and certain level journals.
Is it two? What we've heard is that there's 4 different compendia and they have different degrees of strictness. I can't remember which one. We were recommended to go to the easiest one first, and we just have to sort of figure that out. But we've got the second publication. We're working on writing it. One of my colleagues got on the phone with people who've been taking it off label and getting a sense of their qualitative experience. So, the quality of the data is not as good, but it's supportive evidence. But we'll start talking to them and see what they need.
Yeah, thank you.
Hi, this question's for Chris Lee Messner. With the nortriptyline, a bunch of our children have gone through gene site testing to figure out what drugs would be best for them. Is that a sufficient analysis to see whether it's safe for our specific kiddos? Do you know, have you heard of gene site testing?
I do not know about gene science. Can you tell me more?
Yeah, so a pharmacogenetic testing.
Oh.
It's specifically for like antipsychotics, anti-anxieties, SSRIs, mood
It would certainly give,
stabilizers.
if it lists the 2D6 pathway and whether or not you're high, low, or middle, it'll be helpful.
Okay, yeah, because I see both my kids have had it done. Nortriptyline is listed there. I just didn't know if that was familiar.
Yeah, no,
safe
so in theory, it wouldn't matter whether or not you're a fast metabolizer or a low or slow metabolizer. We would just be titrating more slowly if you're a slow one.
Okay, thank you.
Great session, thank you. For Dr. Lee Messer, dose optimization is a really thorny topic. I mean, clearly for the girls who had KCNT1, hydrocephalus became a sign that they had crossed the NOAEL. And for your effort with nortriptyline, are you writing in dose-limiting toxicity criteria? Could you be more specific and expand on that topic?
Yes, I'd be glad to. So we've talked about a lot of this, and again, we're using somewhat the local doctor to help make that decision. So, at least in my case, I've used nortriptyline or amitriptyline many times, and so, the response that you usually hear is, Johnny's too agitated. We need to decrease the dose, or it was doing great on 10 milligrams, but now on 15, it's not looking good. So it's not a question of like in these cases that toxicity is not a safety issue for the most part. Usually it's one of the other side effects that's limiting. I'm sorry if I can indeed that. As part of that sort of template that I've been following, I have been sort of listing larger numbers, and I imagine that our IRB will be asking some of these questions too. But we are, it is a medicine where, by experience, that's sort of the things that are limiting.
Thank you.
Got a couple of questions. Both could be answered by either of you that have talked about clinical trial design. First question is enrolling individuals with Syngap missense mutations that might be classified as variants of unknown significance. What are your feelings about that? And then I can ask my second question after that.
So, in those cases where we don't have a verified one, in general, from the point of view of the IRB and the sort of greater picture of things, usually that's not someone that we would want to put in our study. Like we would probably get in trouble for that because of this issue of we have this evidence that says you should be helped. On the other hand, it could be that we're going to have a registry because it's a fairly safe medicine and it's something that could be monitored with your own doctor. So as of now, I haven't included that, but you can talk to me and, as you can hear, it's an evolving story in terms of whether or not we should, yes, definitely include this, or is there some pre-test that we can use to assess, we saw today like nitric oxide might be a way to assess if Syngap levels are low. So if there's some additional evidence that we can say that we think this is going to help you, we'd be, have a better argument for including it in the study.
I want to piggyback on that
Mhm.
and add that what we found with our study is that we wanted flexibility. And so the IRB allowed us to have language to say, if something like, has a pathogenic mutation or a variant and a consistent clinical picture. And that kind of language has given us flexibility. We didn't have to make those calls in our enrollments. It was pretty clear. But depending on sample size, what we've learned is that, clinical trials, like they're hard. And so, and there's a lot of regulatory burden. And so we built in language to give us that flexibility, so that if we found someone who really clearly met and might benefit, but that the variant maybe hadn't been properly characterized, that we would have that flexibility.
My other question is about study design. We heard a lot about N-acetyl-L-leucine. The clinical trial in Niemann-Pick disease used an N-of-1, aggregated N-of-1 crossover design trial. I haven't heard about that design strategy. Has that been discussed for some of the things that you guys are working on? And what are the pros and cons of that?
So I think I only have half your answer. So I've looked now at quite a few different approaches to trials. And indeed, one of my hopes is that we'll be able to do these more efficient methods in order to make it so that we can do clinical trials across, populations which are very limited, adaptive approaches and things like that. I know specifically, I imagine our stage 2 of to be randomized control trial with placebo, but with a crossover design, sort of as you mentioned, so that we can get sort of the best evidence we can, but everyone eventually will get the medicine.
In the challenge with crossover designs is that you have to take someone who's on the drug for whom it may be working and then take them off. I've struggled with that for epilepsy and seizures because of SUDEP risk. So part of our reason for doing open label was that we had no idea what to expect. And so we just wanted to get a sense. But as we've thought about study designs, the idea of crossover has been suggested, and I've personally been uncomfortable because it's hard to tell a family with a child who's had a lot of seizures and you finally fix them that you're going to break that and take the drug away. I don't have to convince the FDA because they've already signed off on this drug. I just need to convince an insurer. And so I think in our situation, we may have an easier time of it, depending, again, on what these compendia say.
Yeah, so I have a question. It could be for any of the 3 investigators. I'm curious, in your preclinical trials or work, is there any benefit to like a synergistic effect if we combine some of the repurposed drugs together, and or is there a potential like for putting a repurposed with a gene therapy approach? And if we see a combination therapy, would that have a better effect?
I can take that. Yeah, that's something we've been thinking a lot about. It's certainly something that's a lot easier to test with flies than with mice or other preclinical models. So, in our screen for Syngap, we found 30 some molecules that had a positive effect, and we focused on one of them. Certainly, we probably can't do all 30 by 30, but there's probably some rationale to try some combinations, and maybe Haley back there will do it when we get back. But also, in terms of thinking about in gene therapy, I think the game in rare disease is we need everything, and some kids will need more than one thing, and some kids will only need one, and I think we need to have everything in our arsenal already. I think that's the answer.
It's in a trial, you sort of want to do one thing, but when a patient comes to clinic, it's like, here's what I got. How many do you want? It's you just keep going until they stop seizing.
This is to Dr. Grinspan. I just wanted to say thank you for everything with the trial. My daughter, Sailor, 7 years old, and started the trial in January. I was having, I don't even know, close to 200, maybe a little bit more seizures every day, falling down, eye rolls. And it's been months, and she hasn't had a seizure. So thankfully, it's been working for us. But we're
Thank you.
So
Thank you
happy.
for enrolling.
We've also seen some cognition improvements. I don't know if it's all because of Ravicti or if it's a little bit of age and Ravicti combined, but we've definitely seen differences, and all of our teachers have brought it up and said they've seen the differences as well. And so we're just really thankful and hope it opens the doors for a bunch of other parents and families.
Thank you. Enrolling in clinical trials is taking a risk. And so to you and to all the families, I am undyingly grateful because to fly to New York and to enroll your kid in the trial it's a tremendous burden for a chance and you don't know if it's going to follow through. We got lucky in that many kids responded, but thank you for enrolling.
May I, please? About the nortriptyline, do you have any idea about your primary endpoint? Because you didn't mention, I don't know if it's clear for everyone, but I imagine it's behavior based on the scales that is not seizures, for sure it's not seizures. Nortriptyline is available for low research settings, it's perfect, it's cheap, and it's safe. We prescribe nortriptyline, and it's a great drug for anxiety disorder. So this is my question. What is your main endpoint?
What's my thinking about what the primary endpoint will be? Yeah. So, I mean, that is one of our purpose for doing the pilot is that we don't know what the primary endpoint is. We can guess based upon, anecdotal, use of nortriptyline that maybe it's going to be primarily, improve socialization or improve expressive language or decreased aggressiveness or an effect on ADHD. And the mice sort of give us some hints that maybe that's the case from Juliet's work, but we don't know. And that was one thing that I sort of learned about the process was that your pilot study is there to tell you, to give you evidence about what you should choose as your primary endpoint. And, and especially because we don't have the default of epilepsy seizure control that is so effective in a lot of large trials.
Okay, and we have our last question.
Is that me? No.
So when, excuse me, when you measure seizures, clinical seizures in Syngap patients specifically, how was it a diary? I mean, the EEG, it's different, but how was that done?
Yeah, and people, it's imperfect. So there's some redundancy in how we do it. In my visits, I ask families like, how are the seizures? And so I've got sort of clinic-like notes in which I'm just providing some narration. And then we've got a red cap survey that goes out each week that acts like a seizure diary. It's not perfect. The FDA has gotten stricter and stricter about what they want. So if this was a regulatory study, we were trying to get a new drug, this would not be enough. But for this trial, which was exploratory, it was okay. And the families are often pretty convinced that there's an effect. What? Not so countable for a lot of cases. The FDA wants countable seizures, and I don't know that these are all countable. So if we were in front of a regulator, we would have a harder time.
I just want to abuse the CEO's prerogative to make 2 points, and then I'll let you guys go. First, thank you all. I'm pretty sure no clinician gets paid extra for doing repurposing. They just get a lot of flack from their colleagues. And so we are grateful to you and to Dr. Clement for all his help. But I just want two things got triggered for me in this. The question about crossover trials. If you remember Dr. McKee's presentation about seizures and what percentage of our kids seize and what percentage of our kids don't. And of those who are seizing, some of them, they're absent seizures and they're not visible, right? So until we get a better answer to the question on endpoints, when good people like Dr. Grinspan have precious drug and call me up and says, Mike, how fast can you get me 10 patients with countable motor seizures? And he's like on the clock. I have to call people like Corey and say, your kid has countable motor seizures. Would you sign up for this trial? And can you do it tomorrow? And in our population, these patients who have countable motor seizures are very burdened by disease, and precious from the perspective of these trials. So I want to thank Peter and Corey and everyone else who took that risk and joined that trial. But in our community, when you start talking about placebo-controlled or crossover, you're not only asking me for my very precious patients with countable motor seizures, but you're asking me to be really inefficient with them. So if there's a way to make sure that we can get them on drug, it is our preference. And I do not need to hear the lecture about how placebo-controlled is the gold standard, Mike. I've heard that 1,000 times. But I'm telling you, in this population, patients with countable motor seizures are rare and precious. And we've already done our damnedest to bring those seizures down. But anyway, point made. Second point, syngap.fund/I2, EYE2. If you are a patient in this room and you haven't signed up for that trial, I'm coming after you, right? So we have funded Tom Frazier for years. He is building an eye tracking tool that is used to score and track our patients on many matrices. It is a beautiful tool. It is a cheap tool. It is a tool you heard Dr. Lee Messer say he has volunteered at a very low, if not negligible cost to be used. Dr. Frazier is exceptional, ask anyone, and I need every single Syngap patient to sign up for that trial, do it at home, and get as much training data as possible so we can validate that tool for trials, because you are seeing in this presentation how tricky this business is. And the question of what we will measure and how we will measure, like that very good question from Brazil, is the question. What are you going to measure? How are you going to show this works? So when we beg you to do syngap.fund/EYE2, it is with good purpose. And now you get a break. Thank you very much. Thank you to the panel.